And I'll just say briefly, she did her bachelor's degree at Harvey Mudd College, her PhD at Berkeley, and postdoctoral work with Robert Griffin. And she's now been at Columbia about close to 30 years as a leader there. Anne is also, I should mention, a member of the American Academy of Arts and Sciences, an elected member of the National Academy of Sciences. So the title of the talk is Signaling in Biological Systems, Insights from NMR. So go ahead, Anne.
I hope you can hear me now.
Yep, but we don't see your presentation yet.
Enter full screen. Let's see if I can share screen 1. How does that look?
It's coming up. There it is.
A guy that was overly kind. You remember many things from my career with rosy glasses, but I would correct only one thing, that you undersell your own role in encouraging and enabling us to do our first assignments. Thank you so much for that introduction. So I am going to talk today about a couple of systems involved in cellular signaling. The first section will have to do with studies of ion channels and some recent development of methods to analyze allostery in signaling, including methods to identify hotspots for allostery and particular interest in carbonyl dynamics. And those are applications to prototypical simple ion channel KesA. And then time permitting, I'm going to talk about some more unorthodox signaling systems. Some molecules with low complexity domains that engender oligomerization, and contrasting two such systems. So before I dive in, I just want to thank so many people, the sponsors, especially Bradley for bringing us together today, and John, and Luciano and Guy for their sustained interest in this quasi-regional, quasi-international, and wonderful spectroscopy group. So thank you all. I'm beginning with a potassium channel. Of course, potassium channels are one of the largest signaling superfamilies, important drug targets, intrinsic membrane proteins that in many ways typify signaling behavior of other intrinsic membrane proteins. Their particular role is usually to return the system to its resting potential after, for example, a voltage spike. And in that role, their mean open time is quite important. And we're going to explore the idea that that mean open time is controlled by an allosteric process. We were interested in obtaining insights about structure, but more so about thermodynamics and dynamics using NMR, and that's what I'll talk about. This is a model system. It's a prokaryotic system that it recapitulates so much of what is true of the eukaryotic pore domains of ion channels, potassium ion channels in particular. So like the other pore domains, it's a homotetramer of a hairpin-like 2 transmembrane helix fold viewed on the side here in the middle. And I'm curious if folks see my pointer when I'm moving it around. Do you see that?
I don't see. Oh, yeah, I see it. Yeah.
Okay,
yep.
Thank you, Garth. And then viewed from above, just emphasizing the homotetrameric aspect. A lot of things that are important about this architecture, first unveiled, of course, by Rod McKinnon around the turn of the millennium. But the thing we're going to zero in at mainly so-called selectivity filter, where presentation of successive carbonyls makes 4 optimized potassium binding sites. So 4 sites and each one sees 4 ligands above and 4 below. It's a pretty beautiful, nearly symmetric system that brings forward ideas that actually predated the structure, the idea that we should see a snug fit around the potassium to compensate the loss of water, the idea that there might be multiple single file sites where the appearance of one potassium on one side of the membrane is to some degree coordinated with the disappearance on the other side to improve selectivity and speed both. So this beautiful, beautiful structure. So the regulation of it is, of course, what's important. It's in its resting state closed due to steric contacts at the tetramer interface on the inside of the cell. And that structure becomes open so that ions can flow by bespoke stimuli, different stimuli for each occasion or each ion channel. And in this prototypical channel, we use nature's simplest ligand, the proton. Karina Namijian, Cornell Weil has identified the major ionizable residues involved in that protonation event. What we're specifically interested in is the well-known fact that these channels, all potassium channels, once opened, spontaneously close, even though the stimulus that opened them remains in place. So the drop of pH is not reversed, and yet the channel has a finite open time. It was also known from mutagenesis experiments, in particular Yellen's lab, that this is not the reverse reaction of opening. Mutations that affect this reclosing type phenomenon are in a completely different portion of the protein on the outer mouth. So we opened on the inside, we're closing on the outside of the cell. And so a lot of hypotheses regarding the outside of the cell and regions proximal to the selectivity filter appeared in the literature. I was struck by data that was from Perozzo's lab that in a detailed way that we don't have time for today, the reverse reaction of this reclosure depended explicitly on potassium ion concentration. And so we got interested in documenting potassium binding by NMR as a first step along a kind of long march to see if we could get insights. So the working hypothesis, which parallels closely work from Perozzo's lab, is that the system begins in a resting state where the steric block on the inside of the cell, so-called activation gate, is closed, whereas the outside of the cell is a selectivity filter preloaded with ions, so it's in a sense open, it's ready to transmit. Activation refers to opening the inside so that, if you like, both gates are now open. Spontaneous closure involves the protein losing its affinity for potassium, so that instead of this Gritius-like mechanism or knock-on mechanism, it now can only hold on to maybe one potassium at a time, and so it would lose its facile transmission. So opening one gate caused the closure of another gate tens of angstroms away. And then in some way that's not fully fleshed out, the system will gradually return to the original state if the opening stimulus is gone. This inactivation, as we saw on a previous graph, is slower than activation. It's almost on the seconds time scale, but does depend on the particular system. So this paints a picture in which the only active species, there may be multiple inactive species, but the only active species is a metastable state. It's an intermediate of an allosteric process. We intuited that the proper membrane environment should be important for a process like this, and we employed as Guy indicated, magic angle spinning, solid state NMR spectra to probe this system. Assignments are hard-earned in this system. These particular data that I show you are Alison Zhang's, a recent graduate of the lab, who's now at Plexion Industries Pharmaceuticals. And these are correlation spectra that come from multiple 3D correlations down the backbone. We could talk about the details, but we are implementing well-known homonuclear and heteronuclear transfers. For example, selective Hartmann-Hahn type transfers are central here. And this is our wiring diagram prepared by Eric Keeler based on his data and Alison's to show some of the ways that we know which atom is which and what the density of information is. What's interesting is that we were able, by manipulating potassium ion concentration and pH, to stabilize multiple states of the system. In fact, the three key players, resting, activated, and inactivated, which have distinct chemical shifts. And for example, this diagram summarizes the differences between activated at low pH and deactivated neutral pH. In other words, resting and activated. So those kind of differences you can see in and near the selectivity filter and the inner helix, where there's a hinge to open, and here near the activation gate. Logical enough. A recent paper that I won't have time to do justice to is a collaboration with Lucy Delamotte at the Royal Institute of Technology, KTH, Royal Institute of Technology in Stockholm, a MD expert. And what happens here is that we interpret the chemical shifts enhanced by MD sampling of conformers near putative structural assignments of interest, and using a range of publicly available chemical shift predictions, for example, ShiftX and Bayesian interference tools to distinguish between different possibilities for the open state. And we come to the conclusion that what we're referring to here as an open state is probably partially open. I mean, not a fully open state. So in the bilayers as proteoliposomes, we're able to trap an open state, but it probably is not very far open. And so we use the chemical shifts to infer that in a study we could talk more about if folks are interested. Varying the potassium and the pH helped us to identify the sites in the protein that serve as reporters of binding. So the vast majority of the assigned states, and even for that matter, peaks that are less firmly assigned, the vast majority are spectators, and they serve only to tell us that our protein has not unfolded. A handful of sites report that the proton has bound at the activation gate and that the protein has adopted this semi-open state. A larger collection report that potassium has bound in the selectivity filter, and most of those are proximal to the selectivity filter, but there are some that are a little bit more distant. We zeroed in on another group of reporters that we refer to as candidates for allosteric interactions. They're multi-state reporters, meaning that their chemical shift does not have two options, but rather three or more, and that they report to us about the combination of the status of the two gates. And we took the point of view that such a site, if it is affected both by potassium binding and by proton binding, that it's structure would depend on both of those events. Its energetics could depend on both of those events, and therefore it may be a candidate to be a hotspot for allosteric coupling. In other words, it may convey information from one side of the membrane to the other. And I would say that the kind of definitional hallmark of allostery in systems that participate in signaling is that the binding of one ion or one ligand here is dependent on the binding at the second site. And so we pursued this in a titration study. We're plotting here the titration of potassium at the selectivity filter. And this is presented as a function of the logarithm of the ambient potassium. And the measurement is a protein-based NMR, plotting the percent of the protein that appears in the potassium-bound form. So that's our titration format. And what we're showing is that with the protons not bound, neutral to slightly basic pH, the affinity is micromolar. When protons are bound, pH 3.5, and the protein opens, the affinity is altered by more than 3 orders of magnitude. Now, this effect is pretty dramatic. It's completely dependent on the lipid bilayer environment. I don't have time to show you all the details behind that statement, but it's strongly dependent on being in a bilayer and the details of that bilayer. We worried a little bit about, I mean, gosh, a lot of things can change if you go from pH 7.5 to 3.5. So we did experiments in which we identified the coupled acid to be the activation gate, something published a few years back by Manasi Bhate in my lab, but also followed up by carrying out an experiment in a mutant that is constitutively open. It's altered at the activation gate. It was prepared and characterized in the lab of Krina Nimijian. And the NMR of this system was carried out by Jiu or Jeff Sun. And that open state, studied at the same pH as the closed state, recapitulates the same idea that the system is predisposed to not binding the potassium. The potassium affinity is shifted by more than 4 orders. And so 2 different ways to open the channel, and both of them lose their grip on the potassium. We think that's a strong endorsement of the central hypothesis that the deactivated proceeds to open or semi-open or activated by protonation, but that is inherently lower in affinity for the potassium. For some reason, a delayed response, an allosteric response to opening is to lose the potassium. And that's the species which now controls the mean open time. That's our hypothesis. So this allostery, like almost any allostery process, you begin to wonder what are the residues that convey the information of opening to the selectivity filter. And we zeroed in on the basis of our NMR shifts, we zeroed in on a cluster of hydrophobic residues depicted in expanded form here, and they sit here at the base of the selectivity filter just above the hinge, logically enough. And I should say some of these residues were also identified in molecular dynamics simulations from Benoit Roux's group. So it was quite edifying to see an overlapping list of residues. Our thought about those residues would be lop it off and see if there's an effect on this allosteric process. And so that's exactly what Yunyao Shu did, whose data is presented here. 2 mutants. The one on the left, I'll present first. This is focusing in on a phenylalanine that interacts with some other bulky residues from other members in the tetramer. And when shaved back to an alanine, about half of the energy in this allosteric coupling apparently disappears. More dramatic was to remove just a methyl group, to take a threonine in this cluster back to a serine. And then effectively, we've gotten rid of all of the allostery almost within our error bars. And the electrophysiology also says we've made a sort of permanently open system. So both of these mutants put some credibility to our hypothesis that it's a clash. It's a clash in the activated complex, which makes this behave like that children's toy, which can only be open on one side at a time. Of course, we love, and Guy really set me up for this, we'd love to see some of the dynamics of this system in action. But having developed the tools to look site-specifically at proteins with magic angle spinning, we want to pursue those dynamics site-specifically and holistically across all of the visible sites. And so this is a combination, the kind of methods for dynamics that folks used to do with static solid-state NMR, combining that with extensively labeled and medium to fast magic angle spinning. This is a kind of challenge of today. Many labs are involved in thinking about how to do that. It's pretty complicated. The particular challenge we assign to ourselves is how to understand a carbonyl, because that's what's in the selectivity filter. And it's a challenge out there that we think hasn't really been met and that will be very rewarding. So the primary thing that goes on when the carbonyl does angular fluctuations is that the chemical shift anisotropy will change in terms of its projection along the magnetic field or any other important axis in the system. And that change in the projection angle would be visible as a number of different observables, and we're thinking particularly about relaxation times. Now, the carbonyl chemical shift values themselves have been studied in a number of labs. Some beautiful data from Ando and co-workers looking at the importance of hydrogen bonding and secondary structure on the backbone of carbonyls. And a number of years ago, we also pursued an analogous question for carboxylic acids, where many model systems could be pursued by quantum mechanical calculations, NMR measurements in a crystalline state, IR, and so on. And I think the rules of how these hydrogen bonds and how the secondary structures affect the carbonyls could probably still be more explored, but have been at least sketched out nicely in those initial studies and others. The thing is, it hasn't really been applied that much to proteins. There's a handful of very beautiful studies, and I'm thinking particularly about Ben Wiley's work when he was in Chad Reinster's group. There's examples of folks probing the carbonyls in proteins, but it's been challenging because of a particular issue of digitizing the carbonyl with the finite spectral window that one has for indirect dimensions at finite spinning frequencies. As the spinners get ever better in the way that Matthias beautifully described this morning, there's an opportunity to start to look at bigger and bigger anisotropic interactions. However, for most of the methods, like the beautiful methods from Rob Tico's group called ROXA, that also happens to mean that you have to turn up your RF until it's 200 and more kilohertz. So we thought about a modification of Rob and Jerry Chan's work where we scale the interaction to fit into whatever spectral window our hardware affords us. And that's been recently reported. It's work by Keith Fritching, Eric Keeler, and Chen-Ming He in my group. So it allows us to look very conveniently at carbonyls and other large anisotropic shielding interactions with kind of ordinary or compromises with hardware. Like, for example, here's carbonyl nitrogen correlation plane for ubiquitin, and into the third dimension for each of these sites, we have digitized the carbonyl KesA tensor. As a remarkable agreement with some work a number of years ago from Bodenhausen's lab, where with over a dozen different solution relaxation measurements, they were able to back out the anisotropy and the details of the KesA tensor, asymmetry, anisotropy, and actually the agreement is quite nice. We think that this new solid state measurement will probably have nice applications for a number of systems, and our first system of interest has been this ion channel, and we're still in the process of fully analyzing those data, but I'll tell you that one of the things that sort of fell out right away is the strong temperature dependence on those parameters. And so that spurred us on along our way to think about the dynamics of carbonyls, particularly in the selectivity filter. This is, by the way, for the activated state that we're probing. So it seemed natural to think about rotating frame relaxation. It's a method that is very insightful on a broad range of time scales, relatively convenient to implement. And from our prior work, we knew that at least under favorable conditions, we had carried out a proof of principle that one could obtain the precise and accurate hop rate, at least for model systems that are well behaved. So we thought it would be a nice way to probe the backbone carbonyl hop rates and angles. And very analogous to solution NMR, we're talking about measuring a rate during a spin lock. That rate, if digitized and determined, could then be plotted as a function of the applied spin lock over a convenient range. And it's really that global set of data, varying both time and strength, that gives you strong constraints on the rate. And a few features that we don't see in solution normally are singularities around the spinning frequency and double the spinning frequency. Now, these are simulated data out of an old study that we did on model compounds, and we did those simulations using Mikhail Restort's wonderful program, Spin Evolution or SpinEv. And you could see a nice review article about solid state dynamics in a very broad frame by one of our speakers earlier today, Matthias Ernst, and also his co-author, Paul Shanda, to see some more details here. But one of the things that's important is that our ability to probe spectral densities could be thought about as operating in a frame inside the magic angle spinning frame, so that what we probe are spectral densities not of zero, but not of omega 1, but of omega 1 plus and minus omega r, so that our spectral density profiles are offset by omega r, and as it happens, because it's a second rank tensor, also 2 omega r. So that could be an opportunity if you move those spectral densities away from zero. That sounds interesting, and you have another parameter to play with, the spin frequency. However, there's a complication that many of us have struggled with, which is that those conditions when omega-1 is equal to omega R or 2 omega R also induce coherent evolution. And this is an effect explored by David Grant many years ago and indirectly employed in lots of recoupling experiments. So you bring back, for example, the KesA interaction when you sit on or near those conditions. So what that leads to is a situation that the true dephasing behavior due to chemical exchange, excuse me, didn't mean to change that. The true behavior here recapitulated with a numerical simulation and the model-free type picture predicting the effective exchange are systematically quite different. The differences are more pronounced as the KesA becomes larger or the spinning frequency lower. So these are simulations, in this case, carried out by Spinach, Ilya Kuprov's program, and in the hands of Eric Keeler, a postdoc in the lab. And this, these differences between model-free and the true behavior, which are dominated by these coherent effects, have been discussed in the literature I mentioned, like in particular, some papers by Alexei Krushelnitsky. So what we decided to do about that was not just to put up with it, but to develop a new spin lock that defeats the evolution of KesA when you're near that condition. And that'll allow us to come closer to these conditions so that we can get more insightful data about the motions. So we replace the simple spin lock with a sandwich of pi pulses and spin locks. And it turns out after we developed this that we realized it closely resembles a family of pulses developed in Niels Nielsen's lab for a completely different application. And he called those RCW pulses, in case you're familiar with those. So we published this pulse sequence a number of years ago, and we've been using it to probe model systems like ubiquitin, as well as our favorite ion channel. And you can see here, what it does for you. In place of these coherent oscillations, which are confusing and confounding, you get something much closer to a monotonic decay, and you don't lose half your magnetization. So it's got a lot of benefits, and you come much closer to behavior that could be fit by model free, which brings back your intuition and some good ideas about how to, start your data fitting. In KesA, we see trends across the protein where interesting elevation for the activated state occurs near the activation gate, as well as the base of the selectivity filter. And these decay profiles seem to fit best to slow or intermediate exchange as opposed to ultra slow or fast. But we're still in the process of identifying the right model and the right statistics and the right protocol for finding unique fits here. So you'll have to stay tuned for the full analysis of the behavior of the molecule. I want to say before changing topics that you could find, if you're interested in this system, additional insights from the work of Mark Baldus, Adam Longa, Marcus Weingarth, Ben Wiley, and others. Lots of beautiful solid state NMR done on these kinds of systems. Now, I think I only have a few minutes for my second topic, so I'll be very brief. But I just want to share with you our enthusiasm and early progress on a very different kind of signaling system. This is, I call it an unorthodox signaling system. These are systems that are beneficial, functional in humans, that appear to operate as high-order oligomers. The first one it comes from a cell death pathway. Instead of apoptosis, we'll be talking about necroptosis, a programmed cell death pathway that maximally elicits immune response. Upstream, apoptosis and necroptosis involve many of the same very famous players in cell signaling, but the bimolecular interaction between RIPK1, a kinase, and RIPK3, another kinase in the same family, commit the cell to a cell death pathway that's really well suited, not for development like apoptosis, but for high infectious load, like high viral load. Instead of a cascade of proteases, necroptosis involves a cascade of these kinases. And we've zeroed in on this particular complex, RIPK1 and RIPK3, which strongly elicits a kind of irreversible descent into necroptosis. And these proteins are globular domain, linker, and then low complexity domains, a sort of proteins whose repetitive use of stuttering Q's and P's and so on immediately clue you in that they might phase separate or they might form amyloids. With the full length protein, and there was data where we knew that in this portion of the protein, the so-called RIM or RIP homotypic interaction motif, this sequence had been identified as a likely dimerization interface. It was Hao Wu, the crystallographer at Harvard Medical School, it was Hao Wu's observation that when co-expressed, these two proteins form rod-like objects right out of E. coli when they're co-expressed at equal levels. And these kind of objects can also be seen in cell culture. And so we became interested in identifying whether that might be an amyloid, as it looks like it may be. So we zeroed in on the typical beta sheet nature of amyloids by carrying out solid state NMR assignments, and indeed the chemical shifts right away confirmed that the region around that rim domain is rigid and beta sheet-like. We only see, whether it's full length or a truncated form, we only see a small amount of the protein right around the rim domain in solid state NMR experiments. And we understand that by thinking about possible motions of the other domains that may be not fast enough or low angle enough to give us solution NMR. They don't show up that well in inept spectra, but they may also not be rigid enough for solid state NMR. So they might be a kind of no man's land, borrowing here a diagram from Daniel Topgard, who kind of illustrates the very important role that dynamics plays in the visibility of NMR signals. So in this system, there's a rigid little nugget in the middle that we began to analyze, and we could find intermolecular quaternary through space contacts using a number of different pulse sequences, but this particular one is a very simple spin diffusion-like experiment that we refer to as DAR. From that and from other experiments, we identified enough contacts between the two molecules to convince ourselves that we were analyzing a heteroamyloid. One of the, it's an idea that's been advanced in many systems, but to get the structure of a heteroamyloid was very exciting to us. And there were not only NMR contacts that hold the two beta strands together in a parallel sense, but also many hydrophobic type contacts that came out of the NMR data that brought two, in our structure calculation, brought two sheets together to form a kind of globular interior. right? So a lot of the soluble residues pointing out on the outside, and a lot of this hydrophobic bulk from this classic IQ-IG motif in the middle, that kind of faces in and forms a sandwich that hides the hydrophobic bulk. Now, we looked at a lot of different possible topologies, and really this is the only one, this alternating in parallel sheets that form two sandwiches. That's the only topology that would converge in cyana calculations with some of the details indicated here. We took this structure and poked at it with mutants and functional data and some X-ray diffraction of a smaller fragment, cross-linking experiments, and it stood up to all of that. So we think it is a reasonable model of the system that forms in human cells. Maybe the most exciting validation of it was to hear from another lab that was working absolutely independently of us that if they replaced their green fluorescent protein in for the kinase, the normally present kinase, that they were obtaining these confocal microscope images with two bright strips and a spine in between them that was dark. And so that helped us really solidify the model that we built around this amyloid core, that it's nature's way to collect the kinases into a very high local concentration in a strip in a way that's summarized with this cartoon. Now, in this system, there are lots of implications and other studies. I would advertise, if you're interested in this, the recent work from Junxia Liu from Shanghai Tech, where she explores the RIPK3 homo amyloid. But throughout human biochemistry and infectious disease, there are lots of other RIM domains. And of course, what we're thinking about now is the possibility that there's really a library of these hetero amyloids to be explored. So we're continuing the work there. I don't have time to do it justice, but I did want to share with you that we look at another protein that has so many similarities. It's globular with functional domains that are designed to bind RNA, and a C-terminal low complexity domain. It's very important in forming stress granules. It was on a list, I think, when Lewis was talking about stress granule proteins. This is one that is truly essential for humans. And we got interested in characterizing it by NMR through our conversations and long-term interactions with Eric Kandel. So this protein, as I said, it echoes many of the features in the kinase that we talked about. And it also has been many times compared with the famous yeast prion protein SUP35. So the expectation was that, like RIPK3, if we expressed this protein, we would find a C-terminal amyloid that would be rugged, hard to take apart, and connect the dots for us in terms of the biology. We can express this in full length. This was the work of Keith Fritching in my lab when he was recently a postdoc here. And the full-length protein oligomerizes, but in this case reversibly. And it forms a scattering suspension. And so that's kind of indicated here. And it's a time-dependent process that can be initiated by agitation. But the question is, are these amyloid fibers that we're forming? They didn't quite look like that by eye, but that's not the right test. And the cryo-EM or stained EM indicates a completely different morphology. We don't know that these are the representative structures, but we see round oligomers, much lower order. The NMR, on the other hand, showed us that 2 of the RRM domains, these RNA binding domains in the middle of the protein, are well ordered and show chemical shifts very, very similar to what Settler had observed in the excise domain in solution. And no visible signal from the C-terminal domain, the low complexity domain. So that suggests for us ordered soluble domains and invisible low complexity domains suggests, doesn't prove, a micelle-like arrangement where the ordered domains, these RRMs, are available for function on the outside, well hydrated, and now these domains are a little bit more like a liquid-liquid separation and have an unfortunate correlation time, so that we're still in a chase to find their actual NMR signals so we can do a better job characterizing them. So that's a stark contrast with not just the kinases I talked about, but other Q and P enriched domains in modular systems. And it raises an exciting possibility that for some of these systems, we may be able to switch between these 2 forms. So these are some of the things we're excited about pursuing going forward. I want to end just by, again, highlighting some very gifted young people. Keith Fritching, who led the charge on TIA, but was also very, very much involved with the work also carried out by Eric Keeler on developing new methods for carbonyls. Those 2 in particular have been really intellectually driving some of the projects I talked about. I also talked about Yunyao Shu's work on the allostery in the thermodynamics of KesA. Those studies advanced in his hands in a way that I doubt very many people could have done. And so thank you all again for this opportunity and for the terrific rest of the lineup today. It's very, very exciting. So thank you.
Thank you, Anne. It's beautiful. And it's so amazing to watch in the field of solid state NMR, going from getting spectra of proteins to initially structures and assignments of simple things to now you're applying it to so many different biologically important systems. So it's really amazing to watch the maturity the field has reached.
Mm-hmm.
So if people would raise their hands with questions, and I could start with one while people are thinking. So one of the great advantages of solid state NMR in studying membrane proteins is you can study quite large systems. And one of the challenges with membrane proteins is, of course, reconstituting into biologically appropriate environments. So KesA, for example, can be studied in micelles, phospholipid bilayers. There's questions of lipids. Can you just give us something of your experience with these different reconstituted systems? Like, for example, the incomplete opening that you detect by molecular dynamics and chemical shift. Is it really that it doesn't completely open, or it just the system that the reconstitution? So maybe just talk about your experience with these things.
Yes, thank you. That's a great question. I mean, it's a big space, right? So we did a little bit of work in detergent micelles in the early days and quickly decided, for a large number of reasons, that the proteoliposomes would be how we would study them. And my biases, for better or for worse, led us to use chemically pure lipids in mixture and try to recapitulate the properties of prokaryotic membranes. Now, you could have gone another path, but most of the data that I show there, we're in a mixture of about 10% anionic head groups and the remainder zwitterionic and a typical chain length and fluidity for prokaryotic membranes. So we're above the membrane transition. That's important. A lot of studies will be below it to capture more signals, but we're above it. So we're doing a little dynamic filtering and getting higher quality spectra and maybe more functional relevance by making that choice. If we had been in a detergent, we would not see the allosteric phenomena that we report here. And we know that from a mixture of close reads on other studies from other groups around the world who've studied this molecule, and some of our own data. So another aspect that you raise is about the degree of opening. I don't know for sure, but I suspect that what you're implying is correct, that the high density of proteins in these proteoliposomes, that in particular, make the proteins behave more like clustered channels. And clustered channels may be altered in the extent of opening and some of the details of activation and inactivation. So an interesting thing would be to look at nanodiscs where you could, choose a size where you get enough lipid, but you're not chock-a-block of proteins. And so we've had a substantial effort in that area, but it's probably not far enough along to really go over the results yet today. But I think that may be right, that, it's not so much which lipids, but the fact that the typical formulation, just for signal to noise reasons, is so high in local concentration of protein, it's almost like a quasi 2D crystal environment, that for some of this stuff, it may be interesting to back off a little bit, put more lipid in and see how that affects issues, just like what you chose, the degree of opening and some of the kinetics around that, yeah.
I think one thing the system is really ready for is a systematic assessment of different reconstitution methods, like you're getting at. But it may well be that the more concentrated clusters is more biologically relevant, and that's what actually happens in cells, and that and it doesn't open all the way in the cells, but it may be that the clustering is more relevant to the biological system.
Mm-hmm, mm-hmm. Yeah, it sounds like a good question to phrase in that way, Guy. Thank you. Yeah.
So I wonder if there are some other questions. I'll ask another one. Maybe people have a little more thought. And this is kind of a bioinformatics-y question. So evolutionary sequence covariance is very valuable, not only for getting contacts or getting structures, but for getting evidence about alternative states, closed versus open states, have different evolutionary covariance contacts, and also allosteric pathways, which seem to the residues involved in the allosteric pathways, the interactions, seem to be very strongly conserved. So you see this as a signature in evolutionary covariance. Have you or your colleagues had a chance to look at that in KesA to see evidence for, of course, the two alternative states, but also to learn something about allosteric pathways?
No, that's an interesting question. I think those kind of covariants could be interesting also in the other systems that I very briefly talked about in the end. That covariant studies you're suggesting could be interesting. Those studies, to my knowledge, have not been carried out on KCSA. There are some genetics studies, but not identifying this particular cluster or allosteric participants. I don't think so. So it's a good thought.
So there are very good servers for Gremlin and EB Fold from Debbie Marks that run these easily. Well, I'm looking for other people who want to ask a question. Okay. Well, if there are no other questions, then I would thank you, Anne, again, and both Louis and Anne for a really great session. I learned a lot in a short time, so thank you very. Oh, I see. Andy Neukoop has a question. Andy?
Hi, Andy.
And I'm revealing that I haven't read one of your papers, but we're also looking and simulating chemical shifts to try to understand how our systems are working. I was wondering what you were using to simulate your chemical shifts to compare to your experimentally determined ones.
Yeah, so I alluded to a study that was actually a while back. That was a study of about 50 crystalline compounds, and the quantum mechanical chemical shift calculations were carried out so many years ago by Julio Facelli at the Utah Supercomputing System. And I don't think the particular choices, about the basis sets and the software would be that relevant today. And I might misspeak if I did that from memory. We're actually probably getting our most important guidance from somebody very close to home for you, David Case, in terms of what we're thinking about doing for our current data on the proteins. So David Case's suite of programs for fragmentation and QMMM and all that. So that would be fun to talk about in more detail with you and him. But yeah, those are quite old studies on the model compounds. Is that what you meant?
Yeah, exactly. That was the direction I was trying to head, and we're already talking to Dave about the other things we're doing. So let's just have to broaden our scope and our conversations with him.
Yeah. But so we're actually contrasting some different options for the QM part of that, as well as for the preparation of the files and the molecular mechanics. And when it's a little farther along, I'd be happy to share that with you in detail. I think, a big deal for the proteins is what you do in preparing along the lines of like assumptions of protonation states and how much dynamics we're including and that kind of thing. Makes a pretty big deal for carbonyls. I think they're less robust with respect to those choices and assumptions than the aliphatics are. So that's the piece of it we're exploring pretty actively.
Yeah, very excited to follow along with your carbonyl work, because we're starting to think about the phosphoruses on our lipids, and if it's going to be even worse than the carbonyls.
Yeah. So it's just the AF NMR that he's developed, and Orca or other things for the QM, but I really think it's that early prep that's going to be where the story is. Prep of the files. Yeah.
So if there are no other questions, I want to thank again our 4 speakers in the morning. Thanks very much, really outstanding. And also thank the organizers for setting up a decent lunch break. Often these Zoom meetings have little or none. So we have a break now. We'll start again at 1:00 p.m. So thank you. 1:00 p.m. Eastern. Thank you.
Thank you, Guy.
Yep, see you all back here. 37 minutes for the afternoon sessions.